if you restore arousal, how can you see if there is also an awareness restoration or not? And this, we really believe since the beginning that.
And the first thing we show is that if you perform deep brain stimulation on the central thalamus when the monkey is completely anesthetized, you will restore.
We all know this paper by Nicolas Schiff and Ali Reza in Nature 2007. I remember I was doing my postdoc at MGH at that time. I was amazed by this paper. Less known is a paper by Coadon, which is a neurosurgeon in Bordeaux. And I think there's also other works in Germany, previous work in Germany, with acute stimulation, the OR, showing the capability of really inducing consciousness or shifting consciousness states. What is new, I think, here is I would say two aspects. First,
welcome to Stimulating Brains.
Hello everyone, this is Andy. Thanks so much for tuning in. Before we get started with announcing today's guest, I wanted to bring up two small things. First, I wanted to express my sincere thanks to Alaa Taha, who is a PhD student in biomedical engineering at Western University in Canada. Alaa has reached out to me to offer help editing the recordings and has done a phenomenal job to produce the last few episodes. This is super helpful and allows me to keep interviewing experts in the field and to run this podcast, which of course is entirely non-profit and hence essentially a hobby of mine. Alaa, thank you. In case you are listening to this and want to become a part of the project in a similar fashion, please let me know. We are always looking for help and to connect the young brains of the upcoming generation with this podcast. Second, I wanted to mention that since a while, a new framework has been born as a sister project to this podcast. Namely, 3 trainees from my lab, Barbara Hollunder, Garance Meyer, and Patricia Svarova, have started to organize a talk series under the same name. The talks are organized on Zoom and for free and feature 2 speakers each month. They have had tremendous talks already by experts such as Phil Starr, Veerle Visser-van-der-Walle, Ludwig Srinzo, Mike Fox, Shan Siddiqui, Haggai Bergman, Kara Johnson, Roxanne Lofridi, Marie Kruger, and others. All recordings are available online at stimulatingbrains.org. To make this talk series special, Barbara, Garance, and Patricia have come up with a new concept. Each month, they pair a senior speaker with a junior speaker to give talks on a similar topic, followed by a joint discussion. Also, the senior speakers devote 10 minutes of their time to talk about how they got where they are now. So if you ever wondered why Mark Richardson ultimately moved full circle from a swamp on a hill to a swamp in Massachusetts, who were the mentors that influenced Phil Starr most, and why, or which was Mike Fox's path to brain circuit therapeutics, tune in and take a look at this talk series. Barbara and Patricia are bright PhD students in my lab in Berlin, and you can follow them on Twitter under B_Hollunder and Tracy Zvar. Alaa Taha is also on Twitter under Alaa_T_. Garance Meyer, who is a postdoc in my lab in Boston, is not on Twitter, but certainly also open for contact. But now to today's episode. I think we have a tremendous topic in front of us. Wait for it. Consciousness. Recently, Jordy Tasserie joined my lab here in Boston after doing a tour de force 5-year PhD at NeuroSpin in Paris, and after a quick stop in Andres Lozano's lab in Toronto. He did his PhD under the supervision of Béchir Jarraya, who is a functional neurosurgeon at Foch Hospital in Paris, a professor of medicine at Paris-Saclay Versailles Saint-Quay University, and a principal investigator at NeuroSpin in Paris. Together, they recently published a paper in Science Advances, in which they awoke non-human primates that were under anesthesia using DBS. Their study has it all: invasive neuromodulation, an animal model, fMRI, EEG, and dynamic resting state fMRI analysis. Together with their joint mentor Stanislas Dehaene, who leads NeuroSpin and is famous for his global workspace theory, they could demonstrate that it is not only possible to increase arousal, but also consciousness using DBS. I think this is a truly outstanding effort and a Herculean PhD thesis and paper. I hope you enjoy the conversation as much as I did, and thanks for tuning in. Again, Stimulating Brains. So, Béchir, it's a great honor to have you on the podcast. At this point, I already will have introduced you formally, so we can start right off with questions. And to break the ice, usually before we get into the science, I always ask about your free time. So, what do you do when not involved in science? Any hobbies or things you like to do?
Well, I actually love opera, so I love. I'm not singing myself, but when I was young, I discovered opera really accidentally. I was a teenager listening to the French TV broadcasting, and there was a magic flute, which is really very good even for kids and teenagers. The famous Mozart opera, and it was a revelation. I mean, I remember. I have other friends coming here, and I stopped playing with the other friends and went listening to this opera. So it was odd for them, but still. And later on, again accidentally, I discovered Rheingold, the first opera of the famous tetralogy of Wagner. And I became a big fan of Wagner's opera, I must say. So. Again, completely by chance. Of course, I appreciate also other form of arts, paintings, impressionists, modern sculptures, seeing ballet from Tchaikovsky, and of course musicals. When I was in the US, I loved to go to Broadway. Broadway musicals is maybe the modern form of opera, let's say. Yeah. So I enjoy it much, and I would add now that parenting is also a new hobby.
Of course, yeah, that takes a lot of time. So I was about to ask, how often do you get to go to the opera? Is it multiple times a year or every month even? Or
well, it used to be at least every month, but I must say that recently parenting activity took over. So not to mention the COVID, of course.
Sure.
But well, I keep hope that it will go later on back to the previous rhythm.
And you live in the perfect city for art, I would say, with Paris. So that's amazing. So what sparked your interest to get into functional neurosurgery? Why that field?
Well, I must say, Andy, that even I could even question myself why I went to medicine, basically, because at high school I really loved mathematics. It was really my favorite class, my favorite theme, but also biology. And I had some hesitation between medical school and engineering school somehow. So for me, the ideal scheme was to combine both, basically, to do something related to medicine, to help also people suffering or discover new avenues in the disease field. And I found that functional neurosurgery was very well suited for that. I remember very precisely my first year of the medical school. I started medical school in Lyon in 1992. And I read a paper at that time about the Swedish team at Lund, Björklund, Lindvall, who pioneered the grafting in Parkinson's disease. For me, it was really, really amazing to hear about the possibility of neurografting of dopaminergic cells coming from fetal tissues into the brain of Parkinson's patients. And somehow it was like, I want to do that.
Great.
Like the idea of rewiring the brain looked so fascinating. for me. I must say that few domains in medicine also are so transversal, interdisciplinary. I love that. Like in functional neurosurgery, it's, well, we call it functional neurosurgery, but it's not. I mean, you're directly working with colleagues, surgeon, neurologist, neuroscientist, neurophysiologist, MDs, PhDs, the PD nurse. That's for me. I love that actually. For me, I hope it's the future of medicine.
The transdisciplinarity, you mean, to work together with, maybe even a field of neuromodulation that involves many specialties, right? Yeah, that's great. So we heard a bit already this, grafting fascination, and I love it when you sometimes have this aha moment and think, this is what I want to do. I think I had that when I saw the first tractography image as well myself at some point, in small, but it didn't put my entire career in there. But I really loved this idea too, and I remember this image. And so maybe going into that, then in your career, who were key mentors or like turning points where you think this was an amazing experience, or this was the best mentor or the most important person in your professional career?
Yeah, there were several key people actually. My first great mentor, for sure, was Michel Jouvet. Michel Jouvet was really one of the greatest neuroscientists of his time, at least in France and Europe. You probably know that in the 50s, 60s, he really was the first to link REM sleep to dreams, and he could destroy selectively locus coeruleus in cats and see the cats dreaming, basically, because he interpreted that as looking after a mouse. Could the cat dream of that?
Great.
Maybe. And actually, he was a professor at Lyon, a professor of what we call experimental medicine. Like, so today we say maybe translational medicine. And it was his last year before he retires, basically. So I was really very fortunate to be in his class. And I discovered that Michel Jouvet first started his career as a neurosurgeon.
Oh,
which
great.
Is, yes, nobody knows about it, but maybe he was not in the right time for neurosurgery, because at that time neurosurgery was still not on the as today, as developed as even today or even 20 years ago. However, he really gives you with his big charisma, the love of neuroscience, obviously. And he also talked about this grafting in the Loon team, etc. And he refers also to Claude Bernard, so the inventor of experimental medicine. So this influenced me a lot when I was undergrad, or was the first years of medicine in Lyon. Then I started my residency in Paris. I moved to Paris from Lyon for the residency. And I was very happy and fortunate to meet with Stéphane Palfy. He's a functional neurosurgeon and a neuroscientist at Henri Mondeur Hospital. For sure, he played an important role in my career because he was the first to show me a concrete way of conducting both neurosurgery and neuroscience, and going from the bed to the bench. And he was, of course, doing functional neurosurgery. He also gave me the opportunity to go to the lab to do a master and then a PhD under his supervision in the laboratory of Philippe Entraille at CEA. So he initiated me to non-human primate models as well of neurodegenerative diseases. So we did a lot of MPTP modeling, monkeys, and we developed together with a UK biotech called Oxford Biomedica, a gene therapy for Parkinson's disease that eventually later on went for clinical trials. So that was for me really an important moment in my life. I must also acknowledge that people like Yves, the famous neurologist at Tasserie, I did a rotation in his department because I knew that I will do my career in neuromodulation, and for me it was important as a neurosurgeon to also to spend at least a rotation where you are away from the OR, but really having the opportunity to understand what is this disease and this fascinating disease. I mean, and with Yves, I think he gives you again with his charisma and his sense of leadership. I mean, anybody that will meet with him will fall in love of Parkinson's disease for sure.
That's great. I've been trying to get him on the podcast actually because Marwan Harris mentioned I should try, but didn't get a reply. So haven't managed yet. But I must.
Please, yes.
That must have been a great fellowship. So that was a rotation only, or did you spend multiple years with him? Or?
Yes, I did a rotation in his department. And then among also my mentors, I would add Wim van Daffel. Wim van Daffel is a neuroscientist, not necessarily very known in the neuromodulation field, but he's a pioneer in cognitive neuroscience and in fMRI, especially.
Yeah. And that was in Boston, right? That was when you went to the Martino Center. So maybe we can highlight that. There was not Paris anymore, but how long were you in Boston?
So I spent 1 year in Boston. But it was a year, days and nights, basically.
It's, so 2 years.
2 years. Sometimes joke of it, because in Wim van Daffel's lab, actually, we had the opportunity to scan overnight and to use the scanner. So it's this really the efficiency of the US system where the scanners are turning almost 24 hours a day. I try to do these colleagues here in NeuroSpin, it's hard to do. And it was really amazing because it was a switch for me, like he was not doing any Parkinson's or stuff like that, but he developed techniques and technology that eventually later on influenced me a lot in my career when I was back to France, basically.
So let's speak about that. I think after coming back from Martino Center, so from Boston to Paris, as I understood, you founded the Neuromodulation Lab at NeuroSpin, which NeuroSpin, is very famous, needs no introduction, but is I think the center with the most MRIs in France. I could probably imagine. So a bit like the Martino Center in the US or Leipzig in Germany or so, with the mission to evaluate brain modulation by, so your lab's mission was to evaluate brain modulation by pharmacological agents or electrical neurostimulation. Can you sum up your lab in a nutshell, like executive summary? What do you do in the lab?
So this is a lab I had really the opportunity to mount back from Martino Center in Boston. In fact, I was fortunate to be candidate and laureate of a French program for young investigators from ATIP Avenir, called from INSERM. So they give you this opportunity with some funding and some few years. And eventually either the lab succeeds and go for the long term or not. So I found it great, actually. It's a risk, of course, but I love also taking some risk. I think it's important, especially when you are young, you have this opportunity to do that. So for me, it was something I learned in Boston from in Wim van Daffel's lab. How can we do fMRI in non-human primates away or anesthetized? And the idea was to first to study consciousness, because when I was back to NeuroSpin, I made an amazing, amazing collaboration with somebody who's very famous in the cognitive neuroscience field, Stanislas Dehaene. And that was the beginning of one of the most fruitful collaboration I had in
my life over the last 10 years. And I hope it will be also the case for the upcoming years. So with Stanislas, we really decided that these models unique in primates, and the ability of doing neuromodulation combined with functional MRI eventually could reveal some new secrets about consciousness. So how can the brain produce the consciousness phenomena? This is a fascinating field, my opinion. Very hot topic in neuroscience now.
Of course, yeah.
As you know, and even in the neuromodulation field, many teams are interested in applying neuromodulation for patients with chronic disorders of consciousness. So we really, the focus of the lab now is to study consciousness, to dissect consciousness mechanisms, and to apply advanced functional MRI technologies with tasks for the resting states and advanced both technologies and analysis techniques to dissect the neural mechanism of consciousness. And this gives you not only a theoretical dissection of how the brain produces consciousness and modulates consciousness, but also why anesthesia does suppress your consciousness, actually. Why, by binding to GABA receptors or anti-glutamate effect in case of ketamine, or mixed receptors bindings, why do you produce this phenomena? I mean, it's when you think about it, it's incredible. Huh? It's a very recent discovery in the whole history of the humanity, in a reversible manner, of course. So that we could actually have some new insights into how anesthesia affect the brain. And starting from that, we used this model that was developed by Lynn Uhrig. Actually, she's an anesthesiologist herself. And she's also my wife, I should say it. Became my wife after finishing her PhD. And we went to ask the question, could we reverse anesthesia effects once we studied a lot how anesthesia could suppress your consciousness? Could we reverse this phenomenon? And that was the opportunity to make a PhD for Jordy Tasserie. Was my grad student, and it was an amazing adventure, for several year adventure, to could we really reverse that by modulating specific nuclei of the brain, of the thalamus, for example, and by doing so, are we reversing all the aspects of consciousness? Because consciousness is a very polysemic word, so we can apply it for the arousal, we can apply it for the higher level of consciousness, the awareness, the perception, or even the metacognition. And it's very difficult actually to do in that field, either with patients and even more with animals. If you restore arousal, how can you see if there is also an awareness restoration or not? And this, we really believe since the beginning that imaging could be a key tool for that.
Amazing. And I think this is, of course, the main topic also we wanted to talk about, which culminated in Jordy Tasserie's and like your Science Advances publication recently, where I think it's really a tour de force. It also, as you said, took around 5 years at least, I think, this work, or at least his PhD. Jordy is with us today as well, so we can maybe also ask him to summarize a bit how, what the effort was, what the project involved. I think just to excite the listeners already. So in a way, you guys managed to restore consciousness with deep brain stimulation to the thalamus in monkeys, in macaques. So, and you used, multiple MRI techniques and invasive neuromodulation. So I think this is really and these were anesthetized monkeys. So it was not a lesion-based disorder of consciousness, but anesthesia. So under anesthesia with DBS, monkeys woke up again, which I think is very, very helpful and very interesting as a concept. So maybe, Jordy, you want to briefly mention your activities during your PhD with Béchir, and then together we can talk more about the study.
Sure. Thank you very much for having me. It's a pleasure. So we lead a comprehensive study in non-human primates when we basically study behavior of the animals, EEG result, and then we move to fMRI. So we had two groups of monkeys. The first one was completely awake as a reference, and the second group was deeply anesthetized with propofol, is a model developed by Lindring, so finely tuned anesthesia at a deep level. And we perform or not deep brain stimulation with different control, both on the amplitude, so low or high amplitude, but also in terms of localization in the centromedian thalamus, which is an antral-laminar nuclei, or in the ventrolateral thalamus, which is not. And the first thing we show is that if you perform deep brain stimulation on the centromedian thalamus when the monkey is completely anesthetized, you will restore arousal. So what does it mean? It means that the monkey literally woke up. He will have spontaneous movements, he will have self-breathing, he will open his eyes, which is absolutely not the case when you are stimulating at lower amplitude on the same contact, or if you are stimulating in the ventrolateral thalamus. But it just gave us an understanding of how deep brain stimulation can restore arousal, but still not awareness. So we also perform EEG during this same condition. And what we show is that the normal delta power significantly decrease under the same conditions that restore arousal in the monkey. And in the opposite, normalized theta and alpha power increase when you perform deep brain stimulation. So overall, the spectral distribution of frequency and entropy increase during high amplitude centromedian thalamus. But again, we still have no idea about the content of consciousness, so the awareness. So we then moved to fMRI this time. And the first idea was just to look at the DBS-induced activity. So when you simply switch on and off the stimulation, what kind of brain activity would you have? So we mapped the whole brain fMRI response. And what we saw is that with low amplitude stimulation centered on the centromedian thalamus, you will activate localized sectors of the prefrontal, parietal, and anterior cingulate cortex, for instance, as well as the midbrain and the cerebellum. Now, if you perform the same stimulation, but in the control in the ventrolateral thalamus, the activation will be much more restricted to the occipital cortex. Now, if you look at the only conditions that awake the monkey, so the high amplitude centromedian stimulation, we saw an amazing broadcast effect in prefrontal, parietal, anterior and posterior cingulate cortex, but also insular cortex, striatum, hypothalamus, or even midbrain and cerebellum. And if you do the same stimulation at high amplitude, but now in the control in ventrolateral thalamus, it leads to more restricted activation only in the prefrontal, parietal, and temporal cortex. So among all the different DBS targets and levels, only high amplitude stimulation centered on centromedian thalamus activates these broad cortical and subcortical networks, which include especially the cingulate cortex.
Wow, this is really amazing. So you controlled for frequency or like amplitude, and you controlled for the target. You did this, I think, in 2 monkeys. Is that true?
Absolutely. So we had 3 monkeys for the awake pattern, and 2 monkeys were implanted with deep brain stimulation.
Interesting.
So we then move to resting state, and we look at how the spontaneous fluctuation of the brain will look like when you perform against this stimulation. And what we saw is that when you are under anesthesia, all the long-range cortical correlation will be vanished compared to awake. So the question was to say, can we restore this long-range bilateral connection? And during low amplitude stimulation, both on centromedian or ventrolateral thalamus, we barely restore some part of it, especially in the prefrontal cortex, but not the long-range cortical connection from prefrontal to cingulate cortex, for instance. But interestingly, when we perform high amplitude stimulation on centromedian thalamus, so again, the same that restore arousal, the same that increase the mean spectral entropy, awareness, we saw that we restore an awake-like pattern in the global neuronal workspace keynote. So the global neural workspace is a theory of consciousness developed by Jean-Pierre Changeleu and Stanislas Dehaene, when we identify different key areas in the cortex that are probably responsible for consciousness. And we performed this analysis of functional correlation. We show that only this kind of stimulation in the central thalamus restores the long-range cortical functional connectivity.
Super cool. So this even ties in the global workspace theory that, as Béchir mentioned, Stanislas Dehaene, among others, developed. So at the center of NeuroSpin. You were not done. You wanted to. Sorry, did I interrupt you?
Sure, because I think what we demonstrate here is just the mechanism of deep brain stimulation centered in the central thalamus. But from all the experiments, we still have no idea about the content of consciousness, what is really the awareness. And to that, it has been demonstrated that there is two biomarkers of consciousness when you use neuroimaging. And the first one is the dynamic resting state. So if you use an unsupervised algorithm to segregate your fMRI signal into different patterns, you will be able to identify, let's say, 7 brain patterns that occur in your brain and how your brain is moving from one to another during 10 minutes of resting states. And what has been demonstrated both in non-human primates and in patients with chronic disorders of consciousness is that when you are completely conscious, you have a rich set of brain states completely independent to the structure. So if you order this brain state according to the similarity with the anatomy, you will see that there is absolutely no correlation. And what we've done here is that we perform the exact same experimental condition, so awake, anesthetized, and anesthetized plus deep brain stimulation. And we saw that only central thalamus deep brain stimulation at high amplitude was able to completely restore rich repertoire of brain states, which was not just the case when you are completely anesthetized. So when you are anesthetized, your brain is shaped to brain state number 7, which is the one which is really tied by the anatomy. So your brain is not flexible enough to jump from one state to another, but you will remain in one specific pattern. And interestingly, it was absolutely not the case with the other deep brain stimulation condition. So again, the same condition restore rich and dependent of the anatomy pattern in dynamic resting state. And just to finish, I know some of you could be a bit annoyed by resting state and how we can denoise this kind of data. So we move to another paradigm, which is an event-related task, and a notable paradigm we call local global. And again, it has been demonstrated both in patient and in animals models that the first level of deviance, so just the tone would break the rule. You can process that even if you are non-conscious, which is what we call the local deviant. So you will hear something like beep, 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 bap. But now if you make something much more complex, which would be the global deviance, when the entire sequence violates the rule, so you will hear beep, 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 bap, beep, 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 bap, beep, 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 beep. This is much more complex to process, and only if you are conscious, you will be able to have this ability to detect this global deviance. And what we show is that if you perform, again, the same central thalamus deep brain stimulation, you will restore these broadcasting effects in prefrontal, parietal, and cingulate cortex when the global deviance occur, which is not the case when you are anesthetized, because all this effect is completely vanished. And so for us, it was really two different orthogonal way of demonstrating a signature of consciousness that is restored by deep brain stimulation centered in the central thalamus, which is what not the case when you perform the control side of the ventrolateral thalamus.
Amazing. So really a very like tour de force PhD and paper, all in one paper. Béchir, thanks for the summary, Jordy. Béchir, did Jordy miss anything, or what would you add?
No, just to give more context. I think Jordy explained very well the paper and the experiment there. Everybody in the neuromodulation field knows that the idea of stimulating the thalamus to restore consciousness is not completely new, actually.
Sure.
We all know this paper by Nicolas Schiff and Ali Reza in Nature, 2007. I remember I was doing my postdoc at MGH at that time. I was amazed by this paper. Less known is a paper by Coadon, which is a neurosurgeon in Bordeaux. And I think there's also other works in Germany, previous work in Germany, with acute stimulation, the OR, showing the capability of really inducing consciousness or shifting consciousness states. What is new, I think, here is that I would say two aspects. First, so far, and also in the monkeys, awakening monkeys with thalamic stimulation was also reported by Yuri Salman group, Redenbaugh paper neuron, and also the group of Emery Brown and the MIT group, Earl Miller and Emery Brown. I think the novelty here is to measure consciousness precisely, and not to look only on arousal effects, the awakening effects. And this was really difficult to say. I mean, how could we really measure awareness in these animals? It's very hard to interact with. And this Jordy explained very well, this signature that we spent already a while on developing them with Stanislas, with Lionel Nakache, and sometimes first in patient, then in the monkey, sometimes first in the monkey, then in patients. That's the virtue of translational medicine, the back and forth. I think this is something very interesting that, in my opinion, even for future clinical trials involving neuromodulation, whatever the technique of neuromodulation in disorders of consciousness, or why not even in acute consciousness disorder and coma, should really involve these measures, these biomarkers of consciousness, because it's the only way to move on and to prove how much are you inducing in terms of which level of consciousness. And the second aspect, I think, which can be generalized even more, is that functional imaging can be a key tool to distinguish your settings, your electrical parameters. I mean, what I personally think is that eventually functional MRI, functional imaging, it could be also EEG, HDEG, etc., could probably play an increasing role in the neuromodulation field. Like in this publication we had in Science Advances, clearly you could see that the fMRI maps vary a lot according not only to the location of stimulation within the thalamus, but also to the setting and the cortical aspect of it. We know now very well that cortical modulation is very important. Or it's deep, but a lot of cortical effects there. Functional imaging could eventually be a way to better understand the mechanisms of DBS and Parkinson's. Well, this Andres Lozano and his group, and also Robert Jesch in also the pioneering work toward that. But we should probably profit from the advancements in technologies of fMRI and in analysis, of course, of fMRI, both resting state and task. And why not guiding even better neuromodulation settings in our already current applications, movement disorders. It may be even more critical for psychiatric disorders, of course. You know very well, I would not convince you, Andy, about the importance of imaging in the neuromodulation field. You're a pioneer in that. Thank you again also for the collaboration we had so far on lead DBS and adapting lead DBS first as a first time
I should
to clinical models.
Yeah, I should maybe mention that you guys you were the first to use LeadDBS in a non-human. So Jordy, you visited us in 2016 at Charity when we gave the first lead DBS workshop, and you were interested in applying that into like localizing electrodes in macaques. Is that?
Yeah, absolutely. So we discovered this amazing tool, lead DBS, that you guys developed, and we were a bit annoyed to say that it was not yet working for non-human primates. So the idea was to extend that for macaques monkey. And so thanks to this collaboration efforts, we bring this extension when we have now the atlases from non-human primates. And thanks to that, we were able to localize the electrodes that we implanted in this monkey for consciousness disorders.
That's really, really great. And it is now, I think, openly available. People can use lead DBS for primates thanks to you guys. And first publication there. I think just as a side note, there was one group that used it in a swine model without any of my interaction. I didn't even, I just saw the paper.
Wow.
And we ourselves published rats, a rat version of lead DBS and used it in rodents. But that was all after your primate model. So you were certainly the pioneers of adapting lead DBS to other animal models, which is amazing, and was a great.
Thanks to you. Thanks to you. Thanks again.
I didn't do much. No. Really great. Yeah.
And Jordy played an important role.
So I guess for the second point, what you're saying, and I totally agree, we have a causal, impact with the stimulation on the brain. We can now measure that with fMRI beautifully because we really know we induce some stimulus and then we see the response, right? We can contrast location, we can contrast different stimulation frequencies and so on. So I totally agree with you there. For the first point that you mentioned, which was that I think, I guess it was quite obvious to you guys that the monkeys would be more aroused, right? They would move, they would open their eyes and so on. That's easy to diagnose, but sounds like you guys were really into consciousness, right? About, are they conscious or not? That was the key or the most important thing for you. So, and then you think that's where imaging really helped because you essentially can't talk to them. It's hard to measure consciousness there. I would maybe love to dive into that a little bit more, but even starting with arousal. So like when you put the stimulation on at the right frequency at the centromedian nucleus, I think, Jordy, I asked you before, the monkeys would have left their cage, or like they would have gone away if you wouldn't like stop them, right? They were so awake in a way or aroused that they would move and look around. Is that true? Can you describe that or paint a picture of it that we really see how big this effect was of DBS? Because they were under anesthesia, right? I think that is very helpful to hear how strong the effect really was on arousal only.
Yes, so again, the monkey was completely anesthetized at a deep level with propofol. And when we turn on the stimulation with the right parameters, we saw amazing effects. So as you mentioned, you have to picture a monkey that tried to escape, was completely sleeping. And then all of a sudden, he opened his eye. He has their reflex, for instance. He will try to understand the surrounding world, have spontaneous movement. And even on the physiology of the animal, you can see that when you are anesthetized, the mean heart rate will decrease or the body temperature will decrease and it regain normal values. So if you're not turning off the stimulation or if the monkey is not paralyzed, it could definitely turn to something potentially dangerous if you are just next to him. So yeah, definitely you have to picture someone that woke up and restore complaint ability to interact with the surrounding world.
And I think that is so important because, I think many of, you could imagine that the DBS effect had something that you could measure in the signal or so, but no, you really saw it, right? The monkey would essentially wake up. But now I guess for the layperson like me, I'm not, familiar with consciousness that well. It could it be, let's say, like sleepwalking? Like, sometimes we do move during sleep, but we're not awake, we're not conscious. So was it a bit like that? Could it have been like that? And then for that, you needed to prove in different forms, for example, with the stimuli, the bip, 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 bop idea, and with imaging that, no, it was really consciousness, or like at least the measures of consciousness were heightened under DBS? Is that a bit how we could picture it?
It's a good that you use the sleepwalking. So it's a way, maybe a metaphor to dissociate the two dimensions of consciousness, meaning the arousal, the vigilance, which is very easy to see. If you look after an accident and you see somebody in his car, you say, well, is he conscious? It means, is he waking? Is he opening his eyes spontaneously, for example? But then that's not enough. I mean, arousal, you can have arousal or vigilance without any perception of external world. That's something important to that now we have very good examples of it. If you take chronic disorders of consciousness, for example, so these are patients we see a lot in neurosurgery after severe head injury, for example, or severe stroke. And some of these patients, thanks to the current medicine and ICU, can survive, actually. But some of them will even open their eyes, for example, 10 days, 2 weeks, when sedation is not there, and spontaneously eyes are opened. But we know very well that it doesn't mean they are conscious, because they may be vegetative. So they open eyes in the morning, they have a cycle, but actually it's really impossible to have any communication between the external world and those patients. And we know that some of them actually have more than that and start to communicate, to perceive the external world. And we call them minimal conscious state, MCS. And the difference is important because and not easy to make clinically. Colleagues from Liège, like Steve Lorez or Adrien Owen was in Cambridge, quantified that and found that, like, with clinical examination, we are mistaken in 40% of patients because of the limitation of the clinical examination for consciousness. When you go to the department of neurosurgery, or people knows very well the Glasgow Coma scale, for example, this is the universal scale for let's say loss of consciousness. It's a very good scale for the acute management of coma, but it's a very bad scale for the chronic disorders of consciousness, of course. And what we should now teach more and more to our residents and etc. and young colleagues is that after the acute period of a coma, the Glasgow Coma Scale don't have any sense anymore. You need to move to specific scales called, for example, CRSR, so Coma Recovery Scale, revised. There are other scales, but CRSR is one of the most translated in all languages. These are scales that will take you more time to realize. You need almost between half an hour and 1 hour, whereas Glasgow Coma Scale in 2 minutes you can easily do it, but they are much more appropriate to try to better evaluate the recovery from coma. But even CRSR is not enough, and there are patients who are in between vegetative also now, although now we say more and more UWS, so unresponsive wakefulness syndrome, versus minimal conscious state. And this difference can be important. It maybe it's important to select the patients for neuromodulation eventually, maybe in terms of prognosis eventually. So there are different consequences. But this again gives you clear, from the clinical experience, clear difference between arousal vigilance. So the first level of consciousness, which is clinically diagnosed by open eyes or not, basically. And going more than that, perceive the external world and eventually communicate with the external world. So having some kind of awareness, even if it's an intermediate sort of part of the awareness. And clearly, it's important for neuromodulation to restore both aspects of consciousness, especially awareness. So we call it also conscious access or conscious content of consciousness. So it's a more cognitive aspect of the consciousness. It's very important to look at the potential restoration of this second level of consciousness, in my opinion, because this is more clinically relevant and useful for patients with disorders of consciousness.
So
this again can be tricky to measure, and more it's tools like imaging with tasks like local global is not the only one. Many people are working on tasks, but as Jordy mentioned, also more and more within the resting state, also biomarkers within the resting states can make eventually the signature and be a key role both for diagnosis and developing therapeutics.
So measuring consciousness, the level is not easy. Seems I've heard of the zap and zip idea, where you zap with TMS and then you measure some sort of entropy of the response.
Sure. This
is that would that be interesting to do with your model as well, where you have a single DBS stimulus or something, and then look at the entropy or like the complexity, I think, of the response?
And the propagation of the complexity actually across the brain. Now, this is an amazing idea developed by Massimini and Tononi. When they compute specific index called PCI, perturbational complexity index. PCI is a very good measure for sure. Actually, the interesting thing with PCI is that the stimulation is pretty potentially pretty homogeneous across patients and is not relying on the preservation of sensory inputs, because by the end, in event-related paradigms, this is also what you do, but instead of doing electrical zap, you will make an auditory paradigm or a visual paradigm. However, you need in these patients with head trauma also to check for the integrity of the sensory input, visual input, hearing, auditory input, etc., etc. I think somehow, I mean, I would say there are two aspects. First, that it's good to have different biomarkers, because probably we will converge to this idea of personalized medicine. And you know how much brain trauma patients are heterogeneous. They are much more heterogeneous than Parkinson's patients, which already are heterogeneous. So probably you need to multiply the biomarkers. And there is also a strong efforts to do for all of us to bring these biomarkers to the everyday clinical settings. And that's really something we need to work on. And I know, for example, the team of Jacques Ossit and Lionel Nakache and Pitié-Salpêtrière and ICM, they are actually developing embarkable devices that can eventually run this with EEG. So some tasks with online analysis. It's a general question now when you have a hot topic in neuroscience, but only very few coming to the patient.
Sure. No, absolutely. I mean, going towards that, next steps on the way to a clinical trial of this, like developing this into patients, but also if you had done in theory the same thing with humans instead of monkeys, I know not ethically possible, but if you had, and they would be awake, like aroused, you could try to talk to them, right? So you have different means of accessing their consciousness just by interacting with them, maybe that go beyond the monkey. And could an intermediate step, because we do implant CM in epilepsy patients, right? We put electrodes into the central median nucleus and the thalamus in epilepsy patients, sometimes under anesthesia. Could that be an intermediate step where one could say, okay, we try to, of course, with ethical approval and everything, but we try to replicate potentially awaking or arousing patients during surgery that go in for epilepsy? I don't know, is that an idea that's feasible or thinkable?
Well, no, you're absolutely right. I think it depends on what is the idea behind, because clearly development of neuromodulation for consciousness restoration is very clearly not only for chronic disorders of consciousness. You're absolutely right. I mean, Blumenfeld showed that in many papers, and he's driving several preclinical and I think also clinical trials to perform neuromodulation (DBS) to restore consciousness in epilepsy patients. And I think he's absolutely right, because some of epileptic patients have so we're talking of course of drug-resistant epilepsy and drug-resistant epilepsy with consciousness alteration. So this is a strong handicap, and I found it a clever way to address also the question by taking it on the consciousness side more than on the epilepsy side. Although fixing the two problems is of course the holy grail, but still, it could be that you're ameliorating the everyday life of patients enormously already by going in the direction of consciousness restoration. And again, I would try to apply neuroimaging in these patients then to guide the way we conduct, we personalize maybe the settings. Well, you know very well, Andy, I mean, in the movement disorders field, who is probably the most mature application of neuromodulation today. And we know how much it's key to get the right patient, the right spot, the right setting, at the right moment. So go extremely toward personalized medicine. And we can guess that this should be even more important in cognitive disorders, psychiatric disorders, disorders of consciousness. These are also network disease. Consciousness is not related to one center. There is no center of consciousness within the brain. And especially this second level of consciousness, we're talking about the awareness of the conscious access. If we take whatever the theory actually, but it's true that at NeuroSpin, we're taken the GNW theory framework, let's say, to explain also our results. In this theory, awareness happens when the sensory information broadcast over several brain areas. This is the difference between, say, you're looking at this apple and it's going unconscious, and you're looking at this apple, but it's taken your consciousness at that moment.
Firing up,
because this visual input that started in V1, V2, until your prefrontal cortex to parietal cortex, realizing kind of ignition. This progressing phenomena, this is what makes an information becomes available globally and becoming conscious. So this is something it's much easier to assess with the functional imaging, for example.
Sure.
Because you may, of course, you're very right to say that with patients we can have the communication aspect, which can make it easier sometimes, but you can eventually also go into intermediate steps, and eventually functional imaging could be very helpful.
Are there concrete steps towards clinical translation of your findings for hospital or at the NeuroSpin? Pursue? Only if you can talk about it, of course.
Sure. Well, currently in France, there's a big national debate about end of life and dignity of end of life and the right of patients to terminate their life if they consider that they're extremely handicapped. So is it really the best period for these patients? I don't know. I must say that it's true that there are ethical limitations. I mean, we see it already that today it's not easy to take participation to the fMRI. It's heavy. It's not really realized routinely, apart from very few centers in the world. So what about patients with chronic disorders of consciousness? Like much more challenging ethically to do such studies. And I think we need to keep that in mind. I mean, it's not straightforward to do in these patients what we are doing in the lab settings with either preclinical models or even healthy volunteers that are generally young students,
Sure.
very willing to spend hours in the lab, jumping from fMRI to MEG, to EEG to HDEG, no matter what happens. That's clear. So there is an ethical limitation, and there is also, I think, a big challenge about the selection of patients.
Yeah.
It took almost 10-15 years since the early days of Grenoble with STN DBS to really achieve the current international standard of selecting patients based on L-DOPA response, neuropsychological testing, MRI, etc., even psychiatric evaluation. It took a while. And this is a disease supposedly more simple than chronic disorder of consciousness, although we know now that it's not that simple with alpha-synucleinopathy theory. So this, I think, today we're lacking that. How would we select these patients? Because it's not very supportable, in my opinion, to just go again and implant leads, centrothalamic, because probably it will not work in all patients, for sure.
Sure.
For sure.
So maybe to tie into that, one big limitation of your model, or like for translation, is that these were again anesthetized, but then most of your patients, that's not, or all of your patients, that's not what they suffer from, right? So that's probably one
difference. It's a big difference. You have normal structure in these monkeys, completely massively altered structure and connectome in the patient. That's very clear, and that we're very explicit about it, even in our discussions, etc.
I know, but I just wanted to open it up again for the listeners,
sure,
because this is the big difference here, of course, between the model and the reality. Yeah.
You're right. So for me, I hope that I think approaches with the connectome analysis with DTI should, of course, be very important in the selection of patients. But still, for me, in ideal world, we should have a way to predict the effect of DBS before implanting the electrode.
Yeah.
Would it be possible? Now more and more in neuroscience, you have this emergent technique, which is called focused ultrasound, that can allow us eventually to do that. So many of the listeners know focused ultrasounds through the realization of thalamotomies, because of the last years, Inside Tech company make it more and more popular to perform thalamotomies with high intensity, high density focused ultrasound, and to perform a much more proper thalamotomy than done 40 years ago, of course, with MRI control, etc., etc. But I'm not talking about this at all. You probably know that focused ultrasound now allow many other things, allow to open brain-blood barrier, for example, but allow also to perform neuromodulation, actually, reversible neuromodulation. So for me, it is a very interesting pathway potentially that we need probably to first study in the lab setting and even in preclinical models eventually. How could we really predict the effect of DBS with completely deep focused ultrasound neuromodulation? That would be fantastic, because that could save a lot of patients from useless implantations, which for ethical situation like this one would be, in my opinion, very important.
So you're saying with 1 or 2 transducers, low intensity focused ultrasound tries to also, mimic the DBS effect by going deep and inducing action potentials or modulating the activity deep there. And you would say this is a great idea to probe, will this have an effect? And then if it does, you could have a permanent effect with the DBS electrode. That's the model you're proposing. I really like that. That's a great idea. And I mean, even with the Insight Tech machine, I think you can also lower the intensity and modulate, and then have really good precision in the MRI to do it. So it's a great proposal there. Yeah, even also for anything we do in DBS, right? Even in psychiatric disorders, and to probe it before we do the actual surgery is a really important avenue for the field. But I mean, for that we have to show that we can predict the effect first, right? So maybe even something like tremor could be a good model where you could really see it first, and there I think we already know that we can test modulate or
I know some groups already started doing that with tremor. So studying more thoroughly the effect of neuromodulation with the Insight Tech machine at subthreshold level of focused ultrasound. So there will be papers in the upcoming periods.
No, absolutely. And I mean,
about it.
We routinely do it in, like, before burning the hole. I think also here in the hospital, of course, we would always have a test sonication. I sometimes wonder though, because we rarely stop then, right? We would usually then burn the hole. So I sometimes wonder, is the first sonication really just a test? Is no neuron really dying there? Or is it already, that finding that threshold is also going to be important, of course. But that's really, really helpful. So we touched upon epilepsy, and we can maybe go into that again a little bit more, but maybe before that, an odd question, but I'm sure people have asked you this before. If we think of your model modulating the connectome somehow with the global workspace and everything, you could do maybe at some point the same with a multifocal non-invasive idea from outside, and you could maybe even reverse it and do the opposite, and with that maybe have a form of anesthesia non-invasively. Is that something you think about, like to essentially put people into sleep with neuromodulation?
Right. Thank you for releasing some ideas that we are about to test in the future, but I 100% agree with you.
Great.
100% agree with you. And, because you're making this point, I mean, we are working on consciousness and its reversal and its induction with anesthetic agents, its reversal, DBS, etc. And very clearly, I mean, you can say, will anesthesia be the same in 10, 20 years than today? Not sure, because when you think about it, I always joke with my very good friends and colleagues, anesthesiologists. I say, well, anesthesiology have this very unique particularity is that most of the training during residency is about fighting against the side effects of the drug.
Sure.
So most of the training is about how to handle with the circulation effect, the heart effect, the lung effect, the respiratory effects. And of course, this is very technically sometimes challenging, and it's very important. And it's very, for the safety of patients, it's incredible. I mean, the safety of anesthesia increased a lot over the last decades. But by the end of the day, anesthesia is about suppressing consciousness. So the ultimate goal should be, how can I induce loss of consciousness without these side effects, and concentrate myself on monitoring consciousness, monitoring depth of anesthesia. This is also something we don't talk about it much, but as one of potential consequences of this research also is to improve and develop monitoring of consciousness during anesthesia, which of course will correspond to millions or billions of people, much more than disorders of consciousness in terms of number of people. But for sure, I really hope that one day we can go to that direction, that neuromodulation, non-invasive, of course, neuromodulation, could even do the opposite and go for a safe loss of consciousness, safe and reversible loss of consciousness, mainly, maybe, hopefully, without the side effects of the drug, the cardiac, the lung side effects, and the circulatory side effects. Absolutely.
Yeah. And the beauty could be the closed loop component of it, where you can measure and induce and like really titrate the exact level one day, maybe in the future, of consciousness. And that brings me to you, and that, maybe moving a bit to your medical practice as a neurosurgeon specialized in DBS for movement disorders. You're also involved, I think, in the Neuropedis project led by David Linden with other European centers, as well as Toronto and Turkey. And there, I think neurofeedback plays a big role in the way you do DBS. Could you give us an overview about that?
Yes, sure. This is a fantastic project led by an incredible person, David Linden, who is really amazing in the neurofeedback field. The idea is that, neurofeedback is the way to project to the patient his own brain activity somehow, and try to let him to influence that, so that he could, for example, reduce beta oscillation eventually, or reduce the propagation of beta oscillation in the basal ganglia and the cortex, etc., etc. And the idea of David is that neurofeedback, who is more and more providing evidence in diseases, psychiatric diseases, of efficiency, actually, could eventually enhance condition of Parkinsonian patients with or without DBS, but including with DBS. I found it very interesting. I found it also very interesting because it enhances and regroup many groups that are interested in combining imaging and DBS. So it's also a way to promote that field, which is evolving slowly, by the way, for different reasons. Of course, ultimately, fMRI-based DBS is not very easy neurofeedback is not very easy to perform unless manufacturers would help to add applications within their scanners. Why not? Another application could come also eventually from the fact that we are more and more now with the new technologies, for example, the Percept neurostimulator electronic and others probably in the future, read the activity and eventually project it to patients to try to have them influencing that activity. Would that really improve the effect of DBS? Why not? But it's pretty fascinating, actually.
Really cool. So you think there's a role for neurofeedback in these closed loop stimulation techniques? Could you draw a picture of how that could work in the future?
So again, I think during the research development, currently it's based on fMRI. Patient could go to the scanner. fMRI needs to say what will we project? What kind of brain activity will we project? So that's one of the unsolved questions that we try to solve during this project, by the way. Probably not the STN activity, because already the artifact of the electrode is not helping. And again, I believe many evidence now show that cortical effects are so strong in DBS that they can be per se the target of neurofeedback. So could it be just projecting the activity of one of these premotor areas to the patient and asking him to reduce it and having even a carryover effect? Or could we, I'm actually trying to push to to use the methods we used in our consciousness studies. So these dynamic resting state analysis, which is seducing somehow. Could it be also a way to perform neurofeedback? This will still take some few years, but
Sure.
again, I believe it can only help. Huh? And in the study, we have also representative patients. I find it more and more interesting in clinical trials to involve representative of patients, so they can attend even scientific meetings we had. And I must say that it's really very good experience.
Absolutely,
Very interesting
yeah.
experience.
I mean, they are the experts, right, in many ways.
Exactly.
So I totally agree with you. Involving patients, even as co-authors on papers, or is really important. Of course, not every patient would be interested, I think, but some would be, and they are really valuable as partners in science. I couldn't agree more. So new large instruments like the telescope have advanced science, and the NeuroSpin Center has now this new MRI scanner at the EasyOld, I think, an 11.72 Tesla MRI. Which novel discoveries do you expect there? And also, is there any application for your work in DBS, maybe even clinical cases, or better targeting? Any thoughts on that end?
Yeah, thank you for bringing that, Andy. So EasyOld is a fascinating project. It's a European project. It was built between Germany and France. It was even co-signed by Chirac and Schroeder
Oh, wow,
a while
long time ago.
Yes, and it took a while, and it came in. Of course, when I heard first time EasyOld, I said, "Where is Tristan?" Because I thought it was very well chosen. Tristan EasyOld is an amazing opera of Wagner, by the way, and EasyOld was a very well chosen nickname for this Franco-German European project. So the magnet is here, it's running. There were images from post-mortem brain, some preclinical images. I can't say more because it's still ongoing. And I must say that we are very lucky because the regulation agency very recently approved the fact that human beings could go into this magnet. We were afraid that it would have taken more time, but the evidence of safety was enough. Was accepted. So probably next fall, the very first humans will go to this very first time such a high field. But then again, it's big science. It's an interesting. It's also for the physicist, it's amazing. Like between both Germany and France, they already released, I don't know how many patents already, how many discoveries in physics just by building up this magnet. So that's already great. Now, what about neuroscience, and what about even clinical neuroscience? This we need to see, because I would ask you how many, for example, examples where the 7T brings something that the 3T doesn't make actually. Today, on the clinical side, there's only one recommendation for drug-resistant epilepsy for the 7 Tesla as a clinical routine examination. It doesn't mean that I don't believe in ultra-high field. Otherwise, I wouldn't have been a NeuroSpin, of course.
You mean the central vein sign?
That.
You were talking about sorry, that was MS. So what's the recommendation, the clinical recommendation for epilepsy?
I think that to evaluate patients before going for neurosurgery for drug-resistant epilepsy now, if you don't see any abnormality within the image at 3 Tesla, then there's a clear recommendation to make 7 Tesla scan, because you can still see lesions that are not seen at 3 Tesla. But I think it's the unique example so far when you have clear recommendation for 7 Tesla. What would come with 11.7 Tesla? One of my personal guess, I hope at least, is Parkinson's biomarkers. to be able to directly see the neurodegeneration, the neurodegeneration with imaging. You know that at 3T we don't see neurodegeneration. At 70, there are already significant work in South Korea and Italy with Positini, some other groups in the world that starts to see abnormality within the nigra, disappearance of nigrosome 1 signal, etc. But we really would like to go and have clear evaluation, see, for example, Parkinson's in these very, very early stages, because for me, the future for Parkinson's disease, I would be provocative. It's not DBS, it's not HIFU, it's really curing the disease. This is what we should go for. So
you're right.
I hope one day my daughters would say, well, my father implanted leads in PD patient. That was history. Now this disease disappeared.
What a crude measure to do that, right? To put electrodes in the brain.
Exactly.
I hope that too. And I think for Parkinson's, there's great hope. I could imagine for some, there will potentially, even if we could cure Parkinson's or, a large proportion of it or so, there could probably still be indications that come up, right? Like epilepsy, like psychiatric disorders. So you will probably, until you retire, have a job, I hope.
We'll see. So, depending hopefully. On age of retirement, which is increasing progressively in France.
Sure, I heard about that.
Makes
Good
sense.
Point. So, but since we're talking about cure, and we mentioned the better resolution with the nigrosome 1 story, I think that's a really fascinating story as well of detecting Parkinson's better. You also, and you mentioned it in the beginning, you co-invented a specific gene therapy for Parkinson's disease, which also went into clinical trials or practice.
Yes.
Can you tell us a bit more about that? I'm absolutely not an expert on it, but would love to hear more.
Well, so this actually was the work of my PhD under the supervision of Stéphane Palfi, and in an amazing collaboration with Oxford Biomedica, biotech in UK. Actually, the idea there was to restore dopaminergic stimulation in a continuous way and not in pulsatile way. And we know that levodopa is still the main treatment of Parkinson's disease with agonists, of course, but the pulsatile stimulation is really not the best way to administer to restore dopaminergic tone, and you end up having fluctuations, dyskinesia. So the hypothesis was to administer continuously dopamine, and second, to do it also locally, because we know that psychological side effects or psychiatric side effects emerge sometimes from the stimulation of dopaminergic receptors that are, for example, in limbic part within the striatum or other dopaminergic receptors. So being local and continuous was really the aim there. And gene therapy is very well suited for that. So this was a really very nice period of my life when you go to completely new thing where DBS was already the main innovation in Parkinson's, and it worked pretty well in PTP models, non-human primates. And then I was fortunate enough to participate also in the phase 1/2 clinical trial as a young neurosurgeon, chef de clinique. And I remember very well the first patient who just came in after 2 months with this picture, I play golf again. It's really,
oh, wow.
Yes, I call him our Neil Armstrong. He's the first guy. He was brave enough to accept to be the first to have a gene modifying vector, which make you a GM forever. Basically, it's not reversible like DBS, and that these are really rare moments. We always we never forget. Basically, now other formulations of the vector were performed by Oxford Biomedica. So then again, it goes to a phase 1/2 clinical trial. I think it's really a very important and very interesting area of research for the future, because eventually it could even be combined with DBS. There's no contradiction there. And contrary to cell therapy, you don't rely on reconnections. Cell therapy is really very challenging because first, it's not just about grafting cells. You need also to make the connection, the connectivity, and connectivity is very hard to do. And second, we know that cells that are grafted can still be receiving the alpha-synucleinopathy, and several cases published showed that grafted cells could die from the remaining alpha-synucleinopathy phenomena. We know very well now that Parkinson's disease is an alpha-synucleinopathy with this prion-like propagation of misfolded alpha-synuclein. So the disease is there. So I found it very interesting, and I hope that in the future, gene therapy will go also to combat alpha-synuclein. To achieve real neuroprotection and not only symptomatic treatment. But this requires also the very early diagnosis. For me, neuroprotection is something that goes on two legs. First, very early diagnosis, and simultaneous administration of neuroprotective agents. And one without the other, it doesn't work, basically.
Because at the time of diagnosis, it's already clinical diagnosis, it's usually already too late, you say?
It is. It has been demonstrated, like Jeffrey Cordover from Chicago, who spent all his career on developing neuroprotection with his famous paper, the science paper on GDNF in MPTP macaques. He wrote several papers on it. I mean, all clinical translation of this fantastic work failed, unfortunately. Growth factors never worked, even with gene therapy. And one strong reason is that we are still diagnosing too late the disease. And he shows that even 2-3 years after diagnosis, most of the nigra almost is gone. So it's always very late. Now there are hope. There are hope on two sides. There are hopes because two very recent papers showed that we can really screen and test alpha-synucleinopathy either in CSF, and even last week, another paper in the serum, SERA, Nature Medicine paper from Japanese and Luxembourg group. This is a tremendous hope to have a biological biomarker. And I believe that imaging at ultra-high field would complete that very nicely to really show the connectome variation. It might be that one day we will screen in the SERA, because it's easier than CSF, of course, for alpha-synucleinopathy, and select those who will go to ultra-high field imaging. And then you could intervene before even any symptoms happen there. While today, this situation is only possible when you are in a family with a genetic dominant disease. And then we can start hoping that in a decade or so, we may treat Parkinson's before any symptom appears. So that's for me the next big thing.
It's a very, very interesting point. I just finished a collaboration with David Charles and Mallory Hacker on this very early DBS. They had a study, I think, a while back that you're probably familiar with, where they implanted the DBS electrodes, at the time of diagnosis, essentially. So with, very early onset on the disease. It was still, of course, diagnosed, so later than what you dream of. But I talked to David Charles, and he said the same thing, that he would at some point, even if we can show DBS has a potentially disease modifying or like slowing the motor progression effect, to potentially at some point implant before symptoms start, if we can screen for it better. So same idea. He also mentioned that usually it's too late for any disease modifying thing. So do you see future of DBS to people that haven't even, have motor symptoms yet, or would you say that's too invasive, and we would need other agents to modify the disease?
I really believe in. I'm data driven basically, so I believe in what science will really prove. Any disease modifying aspect or therapy, and the most effective one would be then the one that I would adopt, and I would participating in this effort.
Sounds good. And in general, future of we have talked a bit about the future of gene therapy that you were talking about in high field, but future of neuromodulation as a whole, what do you see as next steps that we could pursue?
So I think the future of neuromodulation, I think we would benefit more and more from all what is developed in the labs, basically. So massively imaging, recordings, translating what already exists, actually. So let me tell you, for example, a paper I liked much, Nature Medicine paper about depression by the team of Eddie Chong and Philip Starr.
Yeah, Stanger's paper, yeah.
Exactly. Showing that we are treating a network disease, so we should record everywhere in the network and do personalized medicine. And we ended up implanting this lady in that nucleus accumbens or in that. I mean, there's no single target for sure. Then the challenge with that is that it's pretty invasive. We cannot imagine to generalize this approach to all patients. It's a beautiful paper. I think it's showing the pathway, but we need to do it in a non-invasive way. Can we replace the recording with fMRI? Can we replace the stimulation by focused ultrasound? If we can achieve that, then we do the same approach non-invasively, then you win. And it may be, if in the meanwhile we achieve to cure Parkinson's, it may be that the field will be dominated by cognitive and psychiatric diseases with these kind of highly predictive approaches, including, of course, AI, because AI will probably play a big role in the selection of these targets with these massive parallel data that will be recorded.
Sounds great. I love the avenue. It's a clear path. We just have to follow it and do our best. So to wrap up, I want to be mindful of your time, but some rapid fire questions that I usually include in the end. How does the surgical theater look like in the future? Would you like, if you could design one for, let's say, your son or your daughters becoming neurosurgeons, what would they have? Does it look the same, or which equipment would you want?
I hope that there will be less and less differences between the OR and the lab, let's say. By saying so, I'm not saying that ethically we can do whatever we want in the OR, of course, but I mean that all the strong development, the technological development, the analysis tools that are already existing in the lab and are going even faster and faster, goes very, very translationally fast to the OR. And instead of taking 10, 20 years of delay. And that's our responsibility, Andy, I think. I mean, we have a foot here and a foot here. It's somehow our responsibility to try to push for this quick translation from the two worlds.
I just interviewed Mike Richardson, and he mentioned that his mentor, Phil Starr, had taught him lucky neurosurgeons are the ones that made the OR the lab. And so he tried that as well. So I agree that there's so much that can be done in the OR to do science, but the other way around, of course, as well, to translate scientific tools into clinical practice to help be better and so on. Great. Any eureka moments that you had in your life where you thought, oh, this is, now I understand this, or this was a great success? Or...
Good question. I remember very well, actually, when I was working on gene therapy during my PhD, the very first animal in which we injected the new viral vector, and I was following him directly myself. And I remember it was like 2 weeks after coming back from the weekend, and wow, is he the animal that I saw 2 weeks ago? He was completely transformed. He was getting and jumping everywhere, looking much more healthy and not Parkinsonian anymore. And that's really something you feel, wow, I'm experiencing something amazing here. Then I would say also later on, when we could do all these experiments with Jordy, with lean, combining the fMRI, the EEG, then even the DPS on top of it, and seeing everything working and have these fMRI maps going out of it. Yes, I felt happy because it was exactly what I was looking after when I was in Martinos and saying, I will be back to France and I would like to do this and this and this and this. So, yes.
Great. And it really sounds like a eureka story when, especially just the monkey waking up and or being aroused and everything you put together there. Amazing how it all came together. It's really a very unique paper, just in terms of the depth you've put on, both of you. And any time, like, I'm sure, but did you ever think this was a waste of my time?
Never.
Never?
No.
Okay, that's why. Because it's always worth it, any experiment you do, or yeah,
I think so. We learn from everything we do. Wouldn't say that.
Because I just want to include also, let's say, the failures a bit, because we always speak about success in these podcasts. So any time where you thought that this didn't go well, or that you would be happy to share, or
many times. But I don't know if it is personal optimism. I mean, every time there was a failure, I had two reactions. First, say, all right, I'm sure there's a way to make it working, so keep going. The show must go on. The show must go on.
Love it.
All right, it's terrific. And sometimes if you have a big failure, if you lose part of your experiment, your setup, if it is not working at all, if you crash a machine, I mean, all of us had something like that. All right. And then later on, when I mounted my own lab and my group, then my second reaction was, stay strong in front of your guys, of your students, of your postdocs. Stay strong. I mean, if you yourself feel like, what do they feel? No, guys, you cannot. I cannot.
Love it. Yeah, that makes sense. Yeah. Okay, great. Advice for young researchers entering the field, either in neuroscience or also neurosurgery. Any tips you would give?
Yes, I would give the same advice. I would tell, this is your century. This is your century. This is your time. I mean, we are in a momentum in neuroscience. Profit from this extraordinary development of neuroscience. I would say be transversal, really be transversal. Take risks. Don't stay, move from your comfort zone. I mean, you are too young to have a comfort zone. Maybe there's never a comfort zone. So take risks and believe in your ideas. Yes, I would say that. The brain is a multi-scale, complex object product of evolution. So yes,
Great
if
time
you
to-
are in
yeah.
one scale, sometimes you can move to another scale. You can study single cells, and then you want to do a fMRI, and then you go to clinics, and you are happy with preclinical models. But yes, do that, and share with other people.
Sounds great. We did talk about the future of the field, but right now, what do you think we have missed opportunities, like things we should be doing, but the field is somehow not doing right?
I have the impression, I'm biased, that we are underusing imaging and MRI. I mean, you know that very well, and would even say fMRI also.
Yeah.
So I mean, I feel bad sometimes when I jump from NeuroSpin to the hospital. I say, it's incredible. I mean, you have all this here in the research setting, and the clinic, we are still looking by the eye on this structure and this structure, and fMRI is not obvious to perform in radiology departments. I think we gain to have more PhDs and engineers in the clinical departments. Something I saw in Martinez Center, I mean, the US and radiology department, there are more PhDs and engineers. At least in France, I can generalize to Europe, but radiology departments and clinical departments are 100% about caregivers, radiologists, technicians, which limits a lot completely the expansion of advanced technology, advanced analysis. So either the manufacturers like Siemens, Philips, Toshiba, etc. I have no conflict of interest. I'm just citing some firms. Either they end up developing a ready-to-use device, like, for example, roughly language area, so that the neurosurgeon will be very attentive when he's removing his tumor, or nothing happens. Or sometimes you have an impressive radiologist who is really interested in this and this, etc., but generally feels isolated if there's a lack of environment. So yes, for me, I think we're missing something because of that.
So moving more PhDs into the hospitals, I really agree with you. In Germany, it's the same thing. Big missed opportunity if you compare it to the US, because here we have a lot of PhD PIs even in the medical system. And I think especially on the PI level, I think in Germany, we of course have some PhD students there, but they are not the drivers that lead to actual change. You need to find positions for long-term for people in the medical system that are not MDs sometimes, just to, add in some creativity and technology from the outside. I totally agree. Despite being a big proponent of the clinician researcher model, I think it's great to do both. I currently don't, but I still think it's a great idea. But I also think it's a great idea to include non-medical personnel long-term, give them long-term perspectives in close to the hospitals or in the medical system. The US does a great job.
Exactly, with real careers, they can become professor, etc. I totally agree.
Yeah. Okay. Great. Interesting. Is there anything we should have talked about that I didn't ask, but that you would have liked to cover? I know this was long, but
wow.
Did we cover everything? Or so then, with that, I want to thank you one more time very much for your time. I promised we would go 1.5 hours. We're a bit over time. I'm sorry about that, but thank you for your time. This was really an amazing conversation.
Thank you so much. Thank you.